Hello, everyone, and welcome to our online lecture series for plant biology. Today, we break away from our discussions about photosynthesis and start to take a look at the different plant tissues, organs, and ultimately some of the different cell types that make plants function in the way that they do. Thus far in the class, we've talked a lot about the basics of a plant cell and the most important metabolic process in those plant cells, photosynthesis, which begins with the light reaction on the thylakoid membranes with our two photosystems, P680 and P700, which are made up of pigments like chlorophyll A and B, or potentially, if you're in the brown algae, chlorophyll C. Those chlorophylls are absorbing light energy, transferring it by vibrating back and forth up until enough energy is accumulated in the reaction center. When enough energy is accumulated at the reaction center, or oxygen evolving complex, with its manganese core, is able to rip apart water to produce oxygen and shoot up an electron to a high energy state. The treadmill of enzymes that take this electron and run it for a bit, like pheophytin, plastoquinone, and all the others we discussed last time, produce an ATP molecule. The electron is then handed off to P700, where it again gets transferred to a high energy state, handed off to a treadmill of enzymes like modified chlorophyll A, phyloquinone, the iron sulfur proteins, ferredoxin, and flavoprotein, that ends up charging NADPH. Those two batteries then head over to our Calvin-Benson-Bassam cycle, where Rubisco hopefully is not performing photorespiration, but instead doing its role of carbon fixation, bringing in a CO2 molecule into the cycle. In step 2, reduction, ATP and NADPH are burned, used up, to make ADP and NADP plus to create a 3-carbon sugar. This process is able to cycle back through the third step of regeneration. And in this way, plants are able to provide themselves with all of the energy necessary to grow. So what is driving that growth? How are plants able to grow? Why do they grow in the ways that they do? Well, ultimately, that is a question about tissue patterning and morphogenesis, or the generation of plant forms. So that's the definition of development. How the different tissues in an organism are patterned, and how it ultimately takes its form. Because you will notice here an image of a jade plant and its growing tip. You'll notice that the form of the plant is present from a very early stage. So how come a plant is basically able to arise in miniature from its growing tip and then just continue to proliferate? Well, this ability to kind of become small or start small and then just expand with all of your tissues and layers already present is a byproduct of what we call the apical meristem and the type of growth that it performs. In order to understand why an apical meristem or tip growth leads to this type of growth, we have to understand the 3 regions that govern apical meristematic growth. They are the region of cell division, where cells are copying themselves to become more abundant, so we have more building blocks, more Lego bricks with which to start. Then we have the region of cell elongation, or the region of elongation, where those tiny little Lego bricks, those tiny little cells, are becoming bigger kind of magical to think about, but imagine if your Lego structure could become more massive because the Lego bricks themselves all of a sudden at the same time started to become bigger on their own. That's the region of cell elongation. And then lastly, the region of maturation. The region where that morphogenesis, that region where specific functions are taking place, where the cells are adopting their different specific functions. Now we have two different apical meristems that we will draw out in greater detail and discuss more in separate lectures. They are the shoot apical meristem and the root apical meristem. And yes, I will crack many a joke about the shoot apical meristem looking quite like Batman. This is specifically the shoot apical meristem for a dicot angiosperm, specifically the coleus plant, which we do have in our greenhouse. They're quite pretty. But this same blueprint kind of applies to all of them, even if they look slightly different. All plants, even if they look slightly different. The region of cell division is always near the very tip. In the shoot apical meristem, the region of cell division occurs at the very tip of growth. Whereas in the root apical meristem, the region of cell division is a little bit behind the tip because of an additional structure that we'll talk about later that takes up space ahead of this region of cell division. The region of elongation is always immediately behind the region of cell division. The region of elongation in the shoot apical meristem kind of follows behind where we have our cell division, and because of the way that it is dividing, you will get growth in this direction because cells here will elongate and push or act like they are pushing the tip forward. So you can imagine that this growth is occurring in a similar way as though you were on an elevated platform. The platform is rising. Not that the tip is copying itself, but because the very tip of the plant will stay in the same place. It's not moving, it's not elongating, but rather the region below it is lifting it up through that elongation. And in the plant root, same thing. The region of elongation is behind the region of cell division. Last but not least, we start seeing maturation happen beyond that, both in the region of the shoot and in the region of the root. So once cells have reached their full size in the region of elongation, they then can begin to specialize and adopt different forms and perform different functions. What this means is that when cells are in the region of cell division, they are stem cells, or cells that can differentiate into any different cell type. And what does it mean to be a different cell type? Well, I always like to try to connect these things to us as humans so that way they really mean something to us. You have different cell types. Your eyes are made of a different cell type than your skin. Your skin is made up of a different cell type than your stomach or your heart. In the same way that you have different cell types performing different roles across your body, so do plants. They all arose from a totipotent, or a cell with total potential, can become anything, a cell that was not pigeonholed into being any particular cell type, and then matured in the region of maturation into their specific forms. So you might ask, why on earth do you want to specialize if a stem cell can just do everything? Well, specialization allows for greater efficiency, greater capabilities. I mean, think about this in an economic sense. How much do we gain by individuals becoming specialized in different trades? If I have to learn how to build a car, drive a car, build a computer, be a doctor, grow my own food, how good am I going to be at any one of those tasks? In today's society, we're fortunate enough to allow for specialization, where folks can become really good at different fields, whether that be farming, medicine, engineering. We all have specializations in order to make society run, in the same way that cells have specializations in order to make plants and really any organism work. You wouldn't want your eye to flex into a digestive organ when you're looking at a turkey. It would not be that efficient at being able to detect light. So we have these totipotent stem cells becoming bigger in the region of elongation and maturing into specialized cell forms in the region of maturation. So what are some of those specialized cell forms? Well, I do like this image from your textbook. A cell will begin as a meristematic cell. Maybe you'll be able to recognize some of these organelles. You have the nucleus with nucleolus there, and then the giant vacuole. And this will look like any typical plant cell, right? Looks like the model from the textbook. As it kind of is an image from the textbook, right? But then you start getting really weird looking cells based on subfunctionalization. You can have cells that traffic food in the phloem. More about those in a second, and for a large portion of the class. You can have cells that transport water, that ultimately end up ending their nuclei, ending their plasma membranes. A cell that purposefully commits cell suicide in order to perform its function. Crazy to think about, right? You also have cells that are structurally supportive, like fibers, or cells that are metabolically active. Looks like the cells that we have in our textbook, the typical plant cell, the parenchymal cells. So you have lots of different cell specialization from these different meristematic cells. And so we need to go over the possible fates. What are the forks in the road that an apical meristem can take? Well, first and foremost, the apical meristem can differentiate into a tissue called the protoderm. The protoderm is your pre-epidermis, your pre-skin. So this is where a cell is not yet specialized into the different cell types that are found in your epidermis of a plant. But from here on out, it will only be able to differentiate into different types of skin cells. So that's the difference between this totipotent apical meristem and the primary meristem for these different plant tissues. The second type of primary meristem is the ground meristem. Now, whereas its name would suggest that it's the meristem for roots because it interacts with the ground, don't think of it that way. We consider the ground tissues of a plant to be the bulk or majority of all of the cells in a plant. So it's basically the flesh. The body of the plant is the ground meristem. And then the third tissue type is called the procambium. And this will be your transport tissues. So in this way, you have the apical meristem differentiating into our three different plant tissues. The protoderm, that will be the protective outside. The ground meristem, that will be the living structural supportive inside. So I'll call this your metabolic cells. And then your procambium, that will be your transport tissues. Now, in terms of differentiation when they reach their final forms, these also have names. The protoderm will specialize into the epidermis system. The ground meristem will very conveniently specialize into ground tissue. And the procambium will either specialize into xylem, that transport water, or phloem, that transport food, or the sugars and nutrients of a plant. And if you're taking a plant biology course, if you need to know anything about plants, it is that xylem transports water and phloem transports food. We're going to talk about these a lot throughout this class. We're going to talk about the specializations that occur within them. So please, I believe this is the first major introduction into these. Never forget them. We will be throwing around these vocabulary terms a lot. So these are our different tissues of the plant. You can compare these to the 4 different tissues in animals. Change color here. In animals, the 4 different tissues are our connective tissue, the stuff that makes up our blood and bones, the nervous tissue that makes up our nerve cells, our muscle tissue that makes up, guess what, our muscle, and our epithelial tissue that makes up our skin. So you have something like, grab the pink here, the epithelial parallel to the epidermis, the muscle, nervous, and in some cases connective tissue making up your ground tissues. And then for transport, the specific connective tissue of blood is similar in function, but very different in form from the vascular tissues, another name for these, the veins of a plant. So the epidermis, the ground tissue, and the vascular tissue in the form of xylem and phloem. So, what are our different cell types? Because, even though these are different tissues, I do want to try to go over briefly what the different types of cells that you might find in each of these different tissues are. First and foremost, we'll start with the epidermis. The protective layer that is usually 1 cell thick and on the outside of the plant. The epidermis has, again, 3 basic cell types. We have the guard cells that make up our stomata. We have the pavement cells that make up the outside, generally, of a plant leaf and other tissues. And then we have the hairs on a plant, called trichomes. And so in the past, when we've drawn the underside of a plant leaf, or looked at it in much more sophisticated images in the textbook, these cells were our pavement cells, and our guard cells were the 2 cells on each side of the stomata. What we haven't drawn, though you will be very familiar with, are trichomes, or plant hairs. And this is what they look like. On this plant here, a cousin of the bell pepper, member of the capsicum genus, you have tons of hairs on it in order to help protect the plant. And here you have a scanning electron micrograph, or a very specialized microscope that allows us to see the surface structure of things on a very minute level, and the trichome of a plant called Arabidopsis thaliana. Now, you may not have heard of Arabidopsis thaliana before taking this class, but I can promise you that you will know so much about Arabidopsis thaliana by the end of this class. Because whereas humans use rats or mice as our model organisms for many of our studies, plant biologists use Arabidopsis thaliana as the plant equivalent of the lab rat. It is a member of the mustard family, so you can think of it as related to things like canola. It's a crucifer in the Brassicaceae, and it just so happens to grow very quickly and very easily in small shelf space. We've even grown it here at West Shore, and you can find it outside in the cracks of cement in the summertime. It's a very common plant, very cool, cool rosette of leaves, and used to study a lot of things. So when we talk about trichome development today, or even floral development later today, you can know that those models of plant growth were developed within this lab rat, Arabidopsis thaliana. Now, a trichome is a very interesting cell type because it is oftentimes one giant cell with many copies of its nucleus, with many doses of DNA, generated through a process called endoreduplication. And I include this as a way to show how you actually can get so much specialization in different cells. Seems like a really overly detailed fact, but I think it helps you internalize exactly how cell specialization occurs. You get these different epidermal cells, whether they're guard cells or pavement cells or trichomes, and then in a cell like a pavement cell, the nucleus stays the same. In a cell like a trichome, that cell will start copying its DNA without performing mitosis. No cell splitting. So it's got multiple and multiple copies of this DNA in its cell. And it starts getting bigger and developing a pointy outside. And ultimately to serve its many different possible functions. These plant trichomes generally are sources of defense, glandular, meaning that they produce different hormones, are different secondary metabolites that help protect, keep the plant from drying out, or even hunt other things, as trichomes are used in a lot of our carnivorous plants to grab a hold of the insects that they need to eat. Now, I mention endoreduplication because it is a fascinating phenomenon that we do see in some animal cells as well. Much more common in plant cells, but for example, endoreduplication is a necessary step in the production of platelets within your bloodstream. So you would not be able to scab over if endoreduplication did not happen in that specific cell line, called megakaryocytes. Totally different class, right? Animal stuff. But hopefully you don't mind me including some animal examples, because I think that most folks are more familiar with animals, even in a plant biology class. You, after all, are an animal. So we have these three different cell types within our epidermal layer, or the epidermis tissue system. The guard cells that make up the stomata, the pavement cells that make up the bulk of the epidermis. Those pavement cells are oftentimes making the cuticle that we've used before, the waxy layer that helps keep plant leaves from drying out. And then the trichome, the plant hair extension that performs a lot of functions, is really unique because of its phenomenon of endoreduplication, and is much more giant than a lot of our other cells. Our next tissue type is our ground tissue. Oops. And just to keep things consistent, like we introduced 3 different cell types in the epidermis, we have 3 basic cell types in the ground tissue. Again, these will specialize even more, but I'm trying to keep things to sets of 3 here, where I can, where it makes sense to at least. So ground tissue will specialize into parenchyma, the tissue layer that is alive and retains some totipotency. So you'll notice that sometimes if you cut off a plant leaf and stick it in the dirt, it can then go ahead and start forming a root. We're doing this in the greenhouse currently, propagating some of our plants, and it's oftentimes because of this somewhat totipotency reserved in the parenchymal tissues. These are your basic metabolic functions, like performing of a whole bunch of different photosynthesis. We have our sclerenchymal tissues, sclerenchyma, which are cells with incredibly thick cell walls. These are structural, and interestingly, they are dead at maturity. Meaning that when they've reached their mature form, they're not even a cell at all. They're the equivalent of a PVC pipe. A PVC pipe that isn't, or rather not a PVC pipe, but rebar. They are the equivalent of something structural, keeping the plant up. They don't serve a purpose of transporting things. They don't serve a metabolic purpose. They don't help keep the plant alive. They don't breathe. They don't eat. They don't do anything because the cell itself is dead. All that's left over is this thick cell wall on the outside. That's sclerenchyma. Those are specifically cells that are dead in the ground tissue layer. That'll matter in a second. Our third ground tissue type is the parenchyma. These are alive and also supportive with not as thick cell walls, but you'll oftentimes find parenchyma directly beneath the epidermis. So these are the functional equivalent of quote-unquote bones in a plant. They're found very near the skin to provide a structural support for the plant itself. So we have images of each of these to help try to connect their roles for you. The green cells that have a lot of chloroplasts performing photosynthesis, well, these are parenchyma. In fact, there is more cell specialization. I don't want to gloss over this, but parenchymal cells that have lots of chloroplasts are specifically chlorenchyma. I do apologize for the profuse tendency of botanists to invent names for specific roles. It does help cell specialization, word specialization. They're very useful in communicating things, but it does make for a higher barrier of entry into the field. It does make it more difficult for a person who does not have that vocabulary in order to make it in the field of plant biology. This is true within medicine. This is true within engineering. This is true within every single field. So I do want to emphasize that part of any science class is that you treat this somewhat like a foreign language. There are words that you need to learn. Fortunately, you've got the grammar down. as an English speaker, understanding my sentences already, you already know how to form sentences, but there are just new words, new vocabulary that you need to incorporate, like chlorenchyma or parenchyma, though chlorenchyma is perhaps more detailed than we need here, so don't worry about that too much. Next up, we've got our parenchyma, the tissue layer directly beneath the epidermis that is supportive. So here in this image, you've got the epidermis, and then beneath that, you've got some of your photosynthetic parenchymal cells here, and then you notice that there are cells in the interim that have these thicker cell walls, more support. And so here you have all of your parenchymal tissues. Directly beneath the epidermis, providing support for the skin, kind of like you'd expect from bones. They are alive, they can repair, they still metabolize, they still breathe, they still need all the nutrients that living cells do, but they are supportive. You'll notice how different these cell walls look compared to the absolute monstrosity of a cell wall that we see within sclerenchymal tissue. All of the red stained cell walls here are sclerenchyma. These things are making up a lot of your fibers in the plant. A lot of the supportive tissue that's incredibly dense, incredibly thick to get through. If you're eating something that has a lot of sclerenchyma, you might even think that you're flossing your teeth at the same time that you're eating, because these fibers are exactly that, fibers. But unlike the parenchyma or the parenchyma, the sclerenchyma are dead. They have no nuclei at functional maturity. They have no plasma membrane. The cell part is gone, and the cell wall is what remains. There are two different types of sclerenchyma. We have our fibers, as you see here, running through the length of the stem, or in some cases, like in the pear fruit, sclerids. These are things that look more like jacks, kind of spiky balls, if you've ever played that childhood game, which I know that that's perhaps a couple generations removed from y'all, but you will be familiar with kind of the pokey shape of a jack. And sclerids are supportive structures in things like fruits, like pears. If you've ever eaten a pear, you'll notice that it has kind of a grainy feel to it, different from that of a crisp apple, right? Well, the grainy feel is derived from the fact that the tissue is supported by these sclerids, these jack-like structures that give it a different texture. So, yet again, on a cellular level, you can see how these different tissue types manifest and even give you a different mouthfeel. I personally am a huge fan of pears. They are quite lovely. The graininess is a plus, not a minus. But then again, I have yet to meet, for the most part, fruit I did not like. Not only fruit that I did not like, but food that I do not like. Quite an enthusiast for food. That's in large part why I actually got into plants, because I like to eat. Growing your food, kind of cool, kind of fun. Gets you more in touch with that. Okay, so little digression there, but these are different ground tissue types. So last but not least, we've got our vascular tissue type. For that, let's see, I'll make us a little space here. Vascular tissue. We'll just occupy this last space here. In our vascular tissue, you have the xylem and the phloem. So breaking our rule of threes, or our goal of threes, because there are only two different types of transport tissues in a plant. The xylem that transports water, and the phloem that transports your food. Xylem is dead at functional maturity, meaning that just like your sclerenchyma that hollow out their cells in order to perform their final function of support. Xylem hollow out their insides in order so that they can best transport water. Because you don't want living things in the PVC pipes in the plumbing of your home, right? Living things would be blocking the water from moving. So in the same way, the xylem has aborted its cell in order to allow for the most water to flow through. There are two different xylem types, a tracheid that is evolutionarily first. So these came around first. They're smaller, and vessel elements. These are evolutionarily second and much bigger. We'll spend more time talking about these later. But as a precursor, plants with vessels can grow much, much faster than plants with tracheids. And that, in large part, is one of the reasons why we see some of the plants that dominate the planet now, like grasses, dominate the planet. Those vessel elements are really good at quickly transporting larger amounts of water. The phloem, in contrast, is alive at functional maturity. It stays alive. But just like with our xylem, there are different cell types here, different cell types that perform this food transport. We will again have a separate lecture for those two cell types, but they are the companion cell and the sieve tube cell. And I don't think I've ever spelled the word sieve tube correctly. So I apologize if this is not its correct incarnation. It's either S-I-E-V-E or S-E-I-V-E. Growing up, I didn't even know how to pronounce that. It's funny, English was not my first language. But I remember my older brother correcting me sometime in college because I thought that sieve was pronounced sevi based on reading it. It wasn't a term used in our household. So when I told my brother that was leaking like a sevi, he laughed at me quite heartily. So hopefully that story will help you remember that the two different phloem types are cell types are our companion cells and our sieve tube cells, not sevi. So here are some of the images of these different cell types. In the xylem, you'll often see these stained pinkish because they have thicker cell walls, kind of like we see in the sclerenchyma. It's oftentimes the same polymer, lignin, that's supporting this. Again, vessels are much bigger and tracheids are much smaller. There's a lot more information in this diagram that you need. This is kind of where the end of botany, the end of this plant biology class, will get to where in your understanding of these different elements. But I might as well throw it out there now so that way you start becoming familiar with it. If you want to take a moment to pause the video and read this image taken from Wikipedia, please be my guest. It elegantly summarizes all of the terminology that you will see for the coming weeks when we discuss these components of water transport later. Now, xylem make for some really cool microscopy because the way their cell wall is built is winding like a spring. And so when you are able to squish xylem under a microscope slide with the help of some hydrochloric acid to break up the bindings between these different strands, you will see these xylem elements as springs. We've done a little bit of this in the classroom, and hopefully we get a chance to do so this semester as well. Xylem look really, really cool, but again, not alive at functional maturity. Vessel elements are absolutely ginormous compared to some of the other tissues in the area. So here we have, again, scanning electron micrographs, or that fancy electron microscope that allows us to see the surface structure of things. So you see the difference in size. So this is with the scanning electron micrograph, and this is the same image under a light microscope. Now in terms of phloem, you'll often see them stained teal on our microscope slides. They again have those companion cells and sieve tube cells or sieve tube elements. Talk more about that later, but again, here's an image from a really cool Wikipedia infographic that discusses all the details that you'll need to have by the end of this plant biology course in terms of phloem. Okay, so now we've discussed our different tissue types. Let's talk about how they develop into tissue layers. What are our developmental layers? If I cut a stem across, what am I expecting to see from the outside in? Because we will take this journey many times in many different ways, going from the outside of a tissue all the way to the inside. So here we have an image of flax. Now, I've elected to use flax here because previously, when teaching this class, one of my students was fascinated with flax. This individual would make their own flaxseed oil to laminate different materials that they would make. You know, it is from flax that linoleum is derived. The oil there is what allows, gives that water protective surface. So I've incorporated the stem of flax here. You too can have an impact on the way the class is taught based on some of your interests in different plants. I encourage that. Now, in terms of our plant layers, what are we seeing from the outside in? Well, the first thing you're going to cut is that epidermis. The single layer on the outside of the plant, protecting it from drying out. Now, once you get a little bit deeper, you will get into the cortex. The cortex is mainly comprised of a lot of parenchymal cells, but you will see some parenchyma near the epidermis, and you will occasionally see some sclerenchyma. But this is your ground tissue for the most part. Now, you can continue to go in, and depending on the different tissue that you're cutting in, you may encounter what is called an endodermis. Though this is specific, frequently, to plant roots. But if you do not encounter the endodermis, like in this stem here, there is no endodermis, you will instead get directly into the phloem. Once you pass through the phloem, you encounter the xylem. And this is critical. From here on out, whenever you think of xylem and phloem, it is xylem on the inside, phloem on the outside. I know you can't see it, but I am currently making movements with my hands to show you that it is xylem on the inside and phloem wrapping it out on the outside. This is gospel when it comes to plant architecture. The water transport tissue is in the middle. The food transport tissue surrounds it. This is true for trees. This is true for daisies. This is true, interestingly, in kind of different ways, for ferns. This is true for lycophytes, plants you haven't even heard of. This xylem on the inside, phloem on the outside, is the way plant veins are built. Now, occasionally, in some plant lineages, you again have more ground tissue on the inside of the xylem. Ground tissue on the inside of the xylem is not called cortex. It is called pith. But pith on the inside of cortex on the inside of the xylem is our pith. Developmentally, it has to arise from different stem cells. That's why we give it a different name. We call it pith, not cortex, because different stem cells create it. Here in this flax stem cross section, number 7 is our epidermis. Number 6 is our cortex. Number 5 are sclerenchymal fibers. Look how thick those cell walls are. Number 4 our teal-stained phloem. 3 is our transportive xylem, lots and lots of water transport tissue, a lot of vessel elements in flax. And then number 1 is our pith. You'll notice that here, the pith itself is not retained. Sometimes you'll get these hollow stems. So pith can be alive, or it can be disintegrated. We'll see it both ways in this class. So that's our discussion on our developmental layers, going in order: the epidermis, the cortex, the phloem, the xylem. Now, in some cases, you will encounter an endodermis, and in some cases, you will encounter a pith. But that's the different layers that you're expecting to encounter when you cut through any plant stem or root. The last thing that we'll cover today is our discussion of flower development. You will notice that many times in this class, I try to connect developmental trends with genes, because I think it's important to try to understand these things so that way we can tweak them. Just like we discussed in photosynthesis, where scientists were changing certain parameters of photorespiration in order to help improve productivity of plants, we can additionally see some of the benefits of our understanding of floral genes. So we need to understand the different layers of a flower. Now, we didn't talk at all about flowers as specific tissues. These are reproductive organs. So they get their own specialized thing. That's why they're not included here in our apical meristem tissue types. They will have all of these different tissue types, but they are an organ developed from those different tissue types. The different structures within a flower, the different repeated structures within a flower, are called whorls, because a flower will repeat layers. Those layers from outside in are sepals, then petals, then stamen, then carpels. Now, what's the role of each of these? Well, sepals are your flower leaves. They perform a little bit of photosynthesis in order to give the plant some sugar as it's developing its flower. Petals, y'all will be familiar with because they are the showy organs that help pollinators come visit. Stamen are the male producing pollen, so these are going to be producing the equivalent of sperm. And carpels are the equivalent of ovaries. These are female, producing the eggs. These are the 4 different whorls of a flower. Now, how, from one meristem, do we go about controlling the genes in order to generate a flower? Well, there's something called the ABC model of floral development. And yes, it was discovered within that model plant of Arabidopsis thaliana. I had no idea at the time, but I was actually taking my class on floral development at UCSD from the individual whose father put together this model, which, whoa, I didn't realize it, but later on when I was reading the paper, I'm reading some papers about this, I connected some dots and felt kind of fortunate to get such a level of detail as this dude was getting instructions from his dad at a very young age about floral development. So it's kind of cool that I happened to learn it from him. And now here I am passing it on to you. Small, small world, right? So, the ABC model of floral development relies on the expression of three different genes: the A gene, the B gene, and the C gene. And when I say A gene, B gene, or C gene, these can be groups of genes as well. They're just classified as A or B or C. And in this floral development, because this structure will repeat, C's in the middle, then B's will repeat, and A's will repeat on the outside, so that way you get this similar flower structure. You've got the sepals, green leaves on bottom, the petals that are then, you can think of this as like a vertical cake. 1, 2, 3, 4, 3, 2, 1. In this way, it's patterned from your apical meristem. See that structure still retained in there? Yeah, it definitely is. The sepals are made by tissues that only have A in them. So if the apical meristem is only expressing the A group of genes, it's going to develop into a sepal. If the tissue is developing if your tissue is expressing your A and B genes, this is how you make petals. If your tissue is expressing B and C, well then, you're going to make stamens. And if the tissue is expressing just C, you're going to go ahead and make your carpels, your female reproductive tissue. And in this way, you repeat the structure, you're able to get this perfectly mirrored flower with each of its 4 whorls: sepals, petals, stamen, and carpels. Well, how do we know this? Well, you might be familiar with some of the products of these different genes on a day like Valentine's Day. If you've seen things like an infinity rose, roses that have just petals on petals and petals, or different flowers that have petals and petals and petals, those are flowers in which the C genes have been shut off, either through crossing or through different stress conditions, or only in laboratory settings do we have them for genetic control, or where we've mutated the C genes. But you can get a flower that has tons and tons of petals if you only express A and B, which makes them really good candidates for our Valentine's Day celebrations. There are 2 other genes. Since the ABC model was first identified, we've identified that there is a D gene that's needed specifically in the expression of carpels. And in order to make a flower in general, there is an E gene that specifies that this organ will be a flower, not a shoot. So you have an E gene turn on to make the whole thing possible to be a flower. If E and A are on, you make a sepal. If E, A, and B are on, you make a petal. If A, E, B, and C are on, you make stamen. And if E, D, and C are on, you make a carpel. So I know that that's a lot of vocabulary, but hopefully this helps connect how gene expression in a flower works, and that way you can start to think about how gene expression might work to determine the different cell fates for our different primary meristems, and how an apical meristem will shift into those primary meristems through differences in gene expression. Because ultimately, gene expression plays a massive role in all of this. And that'll do it for our lecture today. I will end the lecture by bringing up this monstrous image of all of the details of the cross-section of a plant leaf and the different cell types that you'll encounter and some of the different structures. I do not expect you to retain this now. This is a tremendous amount of information that we'll go over for a large portion of the class. But in case you want to give it a quick skim, you're more than welcome to. You will be hearing a lot of this as we go. See y'all next time.